Today I want to talk about male reproductive system. As you see in the first picture, in the first slide, we have sperms. The sperms are the male sex cells which can join with the oocyte in the female reproductive system, and they can make the zygote. I will talk about the female sex cells next week for you. Now, you need to know that in the male reproductive system, we have different organs. These organs include testes. We have two testes which can produce sperm and they can secrete hormones. We have some duct system like epididymis, ductus deferens or vas deferens, ejaculatory duct, and urethra. They can transfer the sperm, and during this transfer, they help the sperm to become mature. We have three accessory sex glands, two seminal vesicles, one prostate gland, and two bulbourethral glands. And these glands can make different secretions, and their secretion can make the fluid part of the semen. Then we have a scrotum and the penis. They are different parts of the male reproductive system. And in this sagittal section, you can see these different parts. First, this is the testis. It's located out of the body in the sac, which we call it a scrotum. The testis should be out of the body because the temperature for spermatogenesis should be about 2 to 3 centigrade degree below than the body temperature. This sac is called a scrotum, which the testis is located inside. After the testis, you can see the comma-shaped tube, which is located on top of the testis. This comma-shaped tube is called epididymis. The epididymis has three parts. The first part of epididymis is called head. After that, we have body, and the last part is called tail. The tail of epididymis continue with a long duct, which we call it vas deferens or ductus deferens. As you see here, vas deferens enter the body, pass both sides of urinary bladder, go to the back of bladder or posterior part of the bladder, and after that, the duct of vas deferens and the duct of seminal vesicle gland join together here, and they make the very short ejaculatory duct. This ejaculatory duct enter the gland which we call it prostate gland. As you see here, we have ejaculatory duct, and this is the prostate. The ejaculatory duct and the prostatic urethra combine together, and they can make the urethra. As you see in this picture, the urethra has three different parts. First, we have prostatic part, then we have membranous part of urethra, the part of urethra which pass through the body wall or urogenital diaphragm, and the last part of urethra is located in the penis, and we call it penile urethra or a spongy urethra. These are the different parts of male reproductive system. The function of male reproductive system is production of sperm and the male sex hormones like testosterone. It can help transport, storage, and assist in the maturation of the sperm. It can secrete seminal fluid, and the penis can help the semen to go out of the body and discharge the semen. These are the different parts of male reproductive system. First, I want to talk about the scrotum. The scrotum is this sac, as you see here. This is the supporting structure for the testes. It consists of loose skin, and we have hypodermis beneath the skin, and it can hang the testes from the root of the penis, as you see here. from the external part of the scrotum, you can see the ridge here, which we call it raphe. This raphe can show us inside the scrotum. We have two different compartments, and when we cut the scrotum, you can see the scrotal septum, which divides the scrotum into two compartments, and in each compartment you can see one testis. The scrotum consists of one smooth muscle cell. This smooth muscle cell is called dartos muscle. Pay attention. Last time in the wall of urinary bladder, I introduced one muscle for you, which is detrosor muscle in the wall of urinary bladder. Don't make mistake. This muscle in the wall of scrotum is called dartos muscle. The other muscle which we have in the scrotum is called cremaster muscle. This muscle is called cremaster. Cremaster muscle is associated with each of the testes, and this cremaster muscle is the skeletal muscle. It comes down from this part which we call it a spermatic cord, and I will talk about this spermatic cord later for you. And this cremaster muscle with the other structures which we have in this part can help suspension of the testis from the body. Here you can see the cremaster muscle in the scrotum. This is, as I told you, this is a skeletal muscle and help suspending of testis in this sac. And you need to know that the location of the scrotum and contraction of these cremaster muscles can regulate the temperature of the testis. Because, as I told you before, the spermatogenesis should be done in 2 to 3 centigrade degree below than the body temperature. Previously, we had a disease which we call it cryptorchidism. Cryptorchidism means that during fetal development, we don't have descending of the testis in the scrotum. I will talk about it later for you. During fetal period, we have a small amount of testosterone secretion, and this testosterone secretion helps the testis to come out of the body and locate in the scrotum. If the testis doesn't descend and if it remains in the body because of the temperature of the body, spermatogenesis cannot be done. And these individuals were sterile. Sometimes this sterility is unilateral because only one of the testes remain in the body, and sometimes it's bilateral and both of the testes are in the body. Nowadays, immediately after delivery, if we have boy, the gynecologist check the scrotum, and if they couldn't find the testis in the scrotum, they make a very small incision and take the testis out of the body and put them in the scrotum. Because if the testis remain in the body, that person become sterile. So it's a very important thing which you need to pay attention. The testes are paired oval glands which are located in the scrotum, and the testis is covered partially with a dense irregular connective tissue which we call it tunica vaginalis. I will talk about this slide in the next picture. If you look at this picture, we have a layer which cover the testis from outside. We call this layer tunica vaginalis. Tunica vaginalis itself has two layers. One layer is visceral and one layer is parietal, which you don't need to find it and you don't need to identify. Deeper to tunica vaginalis, we have a white tough connective tissue or a capsule which covers the testis from outside. We call it tunica albuginea. The tunica albuginea become thick in one side of the testis. This thick part is called mediastinum of the testis. And in this mediastinum, from this, mediastinum, we have some fibers which come out and make some compartments inside the testes. These compartments or these rooms, which you can see here, are called testis lobule. And all of these valves which divide this testis lobule are called septa. In each testis lobule, you have 1 to 4 very coiled tube, which we call them seminiferous tubules. And the seminiferous tubules are the tubules which are very important for making a sperm. A spermatogenesis should take place in these seminiferous tubules. First, I want to talk about this tubule and what happened in this tubule, and after that, I will talk about the duct system after these seminiferous tubules. So, up to here, you know that the testis is covered by tunica vaginalis from outside. After that, deeper to tunica vaginalis, we have tunica albuginea. Tunica albuginea become thick in one side of the testis. We call it mediastinum or testicular mediastinum. And from this mediastinum, we have some fibers which come out. These fibers can make septa and make compartments inside the testis. In each of these compartments, we have very coiled tubules. The number of these tubules in each room is 1 to 4. They are seminiferous tubules and they are responsible for making a sperm. In this picture, you can see the lateral view of the testis. Here is the testis, and I told you that the testis is covered with a very tough white membrane or white capsule, which we call it tunica albuginea. And out of tunica albuginea, you have the other layer, which is tunica vaginalis. Between tunica albuginea, and we have 2 layers for tunica vaginalis. One layer is visceral, one layer is parietal. The visceral layer attached to tunica albuginea, and the visceral layer is a little, the parietal layer is a little outside, and between them we have a small amount of fluid like the other membranes which we have in the body. And here you can see the testis, and on top of the testis you have epididymis. As you see in this picture, it's a comma-shaped tube. We have head, body, and tail of epididymis, and after tail, the vas deferens start. The vas deferens start to come up. First, it's located in a cord, in a structure here, which this structure, we call it a spermatic cord, and the spermatic cord contain different parts. I will talk about them later for you. And this is the sagittal section. In sagittal section, you can find some lobules, some compartments, and in each lobules we have seminiferous tubules. Here you can see the microscopic structure of the testis. In this microscopic structure, you can see the tunica albuginea. Here is the cavity of the tunica vaginalis, and this is the parietal layer of tunica vaginalis. And in this picture, you have these cross sections. Each of these cross sections is the cross section of one seminiferous tubule. In this seminiferous tubule, the process of spermatogenesis takes place. Between these tubules, you can see some cells here. These cells are called interstitial cells. In this part, we have many blood vessels, and the blood vessels and interstitial cells together can make interstitial tissue, which I will talk about them later for you. So in this histology, you can see the tunica albuginea, you can see the cross sections of seminiferous tubules, and between these seminiferous tubules, you can see a group of cells. These group of cells which you have here is called interstitial interstitial cells. These interstitial cells with the connective tissue there with the blood vessels can make interstitial tissue. Now, I want to talk about this seminiferous tubule itself. This seminiferous tubule is covered by the areolar connective tissue or loose connective tissue from outside. And you can see a stratified epithelium in seminiferous tubule. This stratified epithelium is composed of two different types of cells. One type of cell is called a spermatogenic cell. They are responsible for making a sperm. And the other type of cell is called columnar sustentacular cell. The other name of this columnar sustentacular cell is Sertoli cell. These Sertoli cells can do supportion, protection, and nutrition of a spermatogenic cell, and I will talk about their function later for you. If you look at this cartoon, this is a part of the seminiferous tubule. In the seminiferous tubule, you have some round cells here. This is the basement membrane or basal lamina, the coverage of each seminiferous tubule. And you can see some round cells here. They are the spermatogenic cell. I will talk about these spermatogenic cells in the next slide. And between these spermatogenic cells, you can see some large yellow cells here. These large yellow cells are called Sertoli cell or sustentacular cell. As you see in this picture, these Sertoli cells can hug these spermatogenic cells. They are much more larger than the spermatogenic cell, and they can protect them, they can support them, and nourish these spermatogenic cells. So this is the picture of a part of seminiferous tube. The sustentacular cells surround the spermatogenic cell. As I showed you before, they extend from the basal lamina to the lumen. Look at here again. They start from this basement membrane or basal lamina and continue to the lumen. These cells are joined together by this junction. These junctions are called tight junction. These tight junctions are very, very tight. I put arrow here. These are tight junction and it's very tight. These tight junctions can make blood-testis barrier. The meaning of blood-testis barrier is the barrier which we have between the blood vessels and the lumen of seminiferous tubule. But why do we need this blood-testis barrier? Think about it, and after I'm done, I ask you need to answer these questions for me. Why do we need blood-testis barrier in the seminiferous tube? The other function which we have for this Sertoli cell or sustentacular cell is helping sperm production. The other function is secretion of testicular fluid. This testicular fluid helps the sperm to swim inside. It's a water, it's a fluid like water which the sperm can swim inside. And also these cells can secrete ABP or androgen binding protein. The ABP or androgen binding protein is a very important protein which is secreted by sustentacular cell. It attaches to the testosterone which is secreted by interstitial cell and keep the level of testosterone in the seminiferous tubule in a constant level, because testosterone inside the seminiferous tubule is important for the process of spermatogenesis. When we go forward, you will understand why we need testosterone inside the seminiferous tubule. Now, I want to talk about spermatogenesis for you. Spermatogenesis means sperm formation. Each day, we have about 400 million sperm production. The spermatogenesis begins at the time of puberty, and it takes place between 65 to 75 days. And we have differentiation and different mitotic and meiotic division in the seminiferous tubules. Now look at this picture. In this picture, you have the testes first. This part, which is thicker, is the mediastinum of the testis. I told you that from the mediastinum, we have some septa which come out and make testicular lobule. And in each lobule, you have seminiferous tubules. In this picture, you have a cross section of one seminiferous tubule, which we make it bigger in this picture. In this picture, you see that the seminiferous tubule is covered by basement membrane or basal lamina from outside. These large cells is Sertoli cell, and the round cells which you see here are a spermatogenic cell. The spermatogenic cells are located and they are in the different rows as you see here. They are like a stratified epithelium, but each layer of the cells have a specific characteristic and a specific type of cell. The cell which is located on the basement membrane is called germinal cell. The germinal cell is called a spermatogonia. The spermatogonia all the times do mitotic division, and by mitotic division, it can increase its number. These cells are diploid cells. The of diploid cell is like the other cells in our body. It contains 46 chromosomes. These cells do mitotic division. The number of the spermatogonia increase and increase. Some of these spermatogonia remain on the basement membrane to do more mitotic division and produce more cells. And the other group of spermatogonia come to the next layer. When it comes to the next layer, it differentiates to a cell which we call it primary spermatocyte. The primary spermatocyte is a diploid cell which contains 46 chromosomes, and this cell starts to do meiosis. You know that the meiosis is a reduction division. By meiosis, we can make the smaller cells which contain half of the chromosomes. We need 23 chromosomes in a sperm. We need 23 chromosomes in the oocyte. When they combine and mix together, they can make a cell which is zygote with 46 chromosomes. So the primary spermatocyte starts to do meiosis. Meiosis has two different steps. First, we have meiosis 1. In meiosis 1, homologous pairs of chromosomes line up at the metaphase plate. Crossing over take place, and we have rearranging of the genes between chromatids. Then the mitotic spindles pull one chromosome of each pair to an opposite pole of the dividing cell. The two cells formed by meiosis 1 is called secondary spermatocyte. The secondary spermatocyte is haploid cell. It contains 23 chromosomes, but these 23 chromosomes, each of them contain 2 chromatids. So we need to do the next step of meiosis, and by the next step of meiosis, we have haploid cell, which contain 23 chromosomes. Each chromosome contains 1 chromatid, and it's ready to make the sperm. I start from the beginning again. Here you have a spermatogonia. The spermatogonia do mitotic division and increase the number. After that, some cells which are produced here come to the next layer and they make primary spermatocyte. They differentiate actually to primary spermatocyte. Each primary spermatocyte starts doing meiosis 1, and the result of meiosis 1 is making two secondary spermatocytes, and these cells are haploid. After that, we have meiosis 2. By meiosis 2, we have 4 spermatids, and they are haploid cells. So from each primary spermatocyte, we have production of 4 spermatids. It's important to know. But you see that the spermatids are round cells. They need to have some changes to make a sperm here. You know that the sperm is a cell which contain a flagella, and these flagella help this sperm to swim. So we need to have some changes in the shape. This change is called spermiogenesis, and I will talk about it in the next slide. All the information you need are here. The spermatogenesis divided into 3 stages. In the first stage, a spermatogonia or germinal cell divide by mitosis. They make 2 types of cells. One cell is type A, which maintain as a germ cell on the basement membrane and do mitotic division again. And the other cell is called type B, which differentiate into primary spermatocyte. Stage 2 is meiosis, and we have 2 steps of meiosis. In meiosis 1, from each primary spermatocyte, we can make 2 secondary spermatocytes, and in meiosis 2, from each secondary spermatocyte, we can make 2 spermatids. So as a result, we have 2 secondary spermatocytes. Each of them can make 2 spermatids, so the result is 4 spermatids at the end from each primary spermatocyte. And now we have a spermatid. These spermatids need to do a spermiogenesis. And by this spermiogenesis, the 4 spermatids need to differentiate into a spermatozoa. Or we can use the simpler word, which is sperm. The sperm shed superfluous cytoplasm. I will show you what happened here. And the sperm contains head, meat piece, and tail. Look at it. Spermiogenesis. We have one spermatid. Our spermatid has a nucleus. It contains a cytoplasm, and the cell is round. In the first step of spermiogenesis, we have multiplication of centriole and migration of centriole into one pole of the cell. You need to know that centrioles are the organelles in the cell which can make a division spin, if you remember from Bio 1010, and also these centrioles can make flagella. So they come into one pole of the cell to make flagella. In the other pole of the cell, we have making some vesicles. These vesicles are made by Golgi apparatus. The Golgi apparatus make these vesicles. These vesicles contain many digestive enzymes. All of these vesicles which are produced here combine together and make a very large vesicle. And this large vesicle is red here, and it's like a cap, it's like a hat. It's located on the nucleus. This vesicle is called acrosome. And and it contains digestive enzyme. This digestive enzyme is for digestion of the wall of our oocyte in female reproductive system, and I will talk about it later for you. Then we have production of mitochondria in the spermatid. We have mitochondria here, and you know that the mitochondria has their own DNA and they can do division and their number can increase. All of these mitochondria come and they are located on the upper pole of the nucleus. I told you centriole start to make flagella, and as you see here the flagella become longer and longer, and then the excess cytoplasm shed off from the cell. Why do we need to remove this excess cytoplasm? Because we want to make the sperm as light as possible. When it becomes lighter, it can swim faster. So this excess cytoplasm comes out, and the excess cytoplasm is phagocytosed by the Sertoli cell. The Sertoli cell can get them, and they can phagocyte this part. And at the end, you can see the sperm. This is the sperm with different, look at this picture. I have one picture of the sperm here. Here you can see the sperm. It contains head and tail. The head of the sperm contains the elongated nucleus. This green structure is the acrosome. This is vesicle, which contain many digestive enzymes. Then you can see a very short part, which is the neck part. After that, you have a middle piece or mid piece of the sperm. In this mid piece, you can see many mitochondria. These mitochondria can make enough ATP for sperm swimming. And after that, you can see the tail. The tail is composed of flagella, and these flagella help this sperm to swim. The sperm is very light and it can swim very fast. So the process which can change the spermatid to the sperm is called spermiogenesis. Again, in this picture, you can see a part of seminiferous tubule. This is the basement membrane. On the basement membrane, you have a spermatogonium. This spermatogonium, as you see here, do mitotic division. By mitotic division, we have two cells. One of these is called type A, and one of these cells is called type B. Type A remain on the basement membrane to do mitotic division again. But type B do differentiation, and by this differentiation, it can make primary spermatocyte. We have differentiation here. When we have our pre-spermatocyte, meiosis takes place. In the first meiosis, in the first step of meiosis, these primary spermatocytes can make 1 and 2 secondary spermatocytes. And in the meiosis 2, each of these secondary spermatocytes can make 4 spermatid. Each of them can make 2 spermatid, and as a result, we have 4 spermatid here. First, these spermatids are connected together by some cytoplasmic bridge, then they separate from each other, and the last step is called spermiogenesis, and spermiogenesis takes, and the spermatid change to sperm. The sperm comes to lumen of seminiferous tubule. The sperm is designed to reach and penetrate the secondary oocyte in the female reproductive system to do fertilization and make a zygote. For this penetration, it needs many enzymes, and I told you that the enzymes are located in the acrosome. The acrosome, which covered the head, contain enzyme to help this penetration. The head of the sperm contains a nucleus, and in this nucleus we have 23 chromosomes. The neck of the sperm contains centriole, and centriole can make microtubules which can make the flagella and the tail of the sperm. The middle piece of the sperm contains mitochondria to make enough ATP for locomotion and swimming of the sperm. And the tail has two pieces. One piece is principal piece and one piece is end piece, and they can help the sperm to move. Here you can see the shape of the sperm again. The head, this is the nucleus, and this is the acrosome. This part is called neck, which we have centriole inside. Here you can see middle piece, which contain many mitochondria, and this is a tail. Tail has one thicker part, which is the principal piece, and one thinner part, which is the end piece. I talked about the seminiferous tubule. Here is seminiferous tubule. Oh, sorry. Or this is one seminiferous tubule, which you can see in this picture. But what do we have between these seminiferous tubules? Between these seminiferous tubules, I told you that we have interstitial tissue. The interstitial tissue contains different types of cells, connective tissue, and blood vessels. Here you can see a blood vessel. This group of cells, which contain pink cytoplasm, are called interstitial cells or Leydig cells. This interstitial or Leydig cell is important for secretion of testosterone. In the next slide, I will talk about it for you. That the secretion of testosterone is regulated by a hormone which we call it LH, and LH is secreted from anterior pituitary gland. The other cell which you can see here is showed by this red arrow. These cells are called myoid cell, and the myoid cell are this one, or you can see the other myoid cell here. These myoid cells are important for making contraction. Myo means muscle. When you have muscle, it means that it contains actin and myosin. Actin and myosin can slide over each other and they can make contraction. When they do contraction, they can contract this seminiferous tubule and help the sperm and the fluid which we have here to move and pass in this coiled tubule. So, between the seminiferous tubules, we have interstitial tissue. In this interstitial tissue, you have connective tissue, you have blood vessels. This group of cells which have a blue arrow are interstitial cells or Leydig cells. They can secrete testosterone. And these cells which are elongated are myoid cell, which can do contraction and help the sperm and testicular fluid to move forward, pass all parts of seminiferous tubules and duct system, and reach the next part, which is didymis. Now I want to talk about the hormonal control of testicular function. At the time of puberty, we have a release of GnRH or gonadotropin releasing hormone from our hypothalamus. Look at this picture. I hope you remember, in our brain we have a part which we call it diencephalon, and in the inferior part of diencephalon you have hypothalamus. The hypothalamus is connected to our pituitary gland by the infundibulum or by this stalk. In our hypothalamus, we can make GnRH or gonadotropin releasing hormone. Then this GnRH come down from this infundibulum and enter the anterior pituitary gland. In anterior pituitary gland, we have different types of cells. And one of the cells which we have here is called gonadotrophs. These gonadotrophs have a receptor for GnRH. So GnRH come and sit on these gonadotroph cells, and these cells start to make FSH and LH. The FSH is the abbreviation of follicle stimulating hormone, and the LH is the abbreviation of luteinizing hormone. First, I want to talk about FSH. As you see here, the FSH can go to seminiferous tubule. It works on all of the cells in seminiferous tubule. You know that we have a spermatogenic cell and sustentacular cell or sertoli cell. If it works on a spermatogenic cell, then a spermatogenesis takes place and can produce sperm. On the other side, it works on the sertoli cells, and the sertoli cells can make ABP or androgen binding protein. This androgen binding protein can attract the testosterone, which is secreted from interstitial cell or Leydig cell in the interstitial tissue. So testosterone come inside this tubule, and the testosterone can promote spermatogenesis. And also these sertoli cells can make testicular fluid. So FSH can promote spermatogenesis, making androgen binding protein and also testicular fluid. When our seminiferous tubules become full of fluid and sperm, the sertoli cells go under pressure. When they go under pressure, they start to release inhibin. Inhibin is a hormone which works on our gonadotroph and do negative feedback. It doesn't let the gonadotroph to make FSH. So by negative feedback, the amount of sperm production and ABP and testicular fluid decrease. So the testicular fluid and the sperms, which are now in the seminiferous tubule, have enough time to pass to the other part of the testis and the other part of the duct system. When these tubes become empty again, we don't have any pressure on the sertoli cells, so the production of inhibin suppress. When we don't have inhibin, again the FSH is secreted. This cycle continue and continue. The other hormone which is secreted from the gonadotrophs is called LH or luteinizing hormone. The LH works on the interstitial cells. It doesn't work on the seminiferous tubules. It works on the interstitial cell or Leydig cell and promote these cells to make testosterone. And this testosterone can change to one metabolite which we call it dihydrotestosterone or DHT. Testosterone and dihydrotestosterone have different functions, which I will talk about it later for you. But pay attention here. Again, when the amount of testosterone and dihydrotestosterone increase in the blood, by negative feedback, this testosterone can inhibit secretion of LH and also it inhibits secretion of GnRH. So all the times in the body of male, we have homeostasis of LH, FSH, testosterone, and sperm production. The testosterone And the dihydrotestosterone have different functions, like male pattern of development before birth, enlargement of male sex organs, and expression of male secondary sexual characteristic, and it can increase anabolism, which I will talk about it in the next slide. The testosterone and dihydrotestosterone, both of them can bind to the same androgen receptors within the nuclei of the target cell. They can work on different types of cells. They can do different functions. One of them is prenatal development. I told you that we need to have the testes in the scrotum. It means that the testes during fetal period should come out of the body and they should locate to the scrotum. This movement take place by a small amount of testosterone, which is secreted during embryonic period. The other function of testosterone and its metabolite is development of male sexual characteristic. They can work on the muscular system and the skeletal system. So you see that the muscles and the bones are bigger in the male than in the female. They can make secondary sexual characteristic in the male, which includes wider shoulder, narrower hip, facial and chest hair, and more hair in the different parts of the body. The skin in the male is thicker than in the female, and the sebaceous gland secretion is more in male than in the female. This testosterone can work on the vocal cord and the larynx of the male, make it bigger, so the voice of the male is deeper than the voice of female. They are the male sexual characteristic. These two hormones can develop the sexual function. It contributes in the sexual behavior, and it can stimulate anabolism or protein synthesis. You know that in the metabolism we have two functions, anabolism and catabolism. Anabolism means making, and protein synthesis in the body of male is more than female. Because of that, the mass of muscles and bones is more in male than female. And as I told you, the negative feedback control the negative feedback system can control the blood level of testosterone. Here you can see the negative feedback loop. When the amount of testosterone increase from this interstitial cell or Leydig cell, it goes to the blood, then it works on the receptor which we have in the hypothalamus, so the amount of GnRH first decrease. When we have decrease in the amount of GnRH, we have decrease in stimulation of gonadotroph cells in the anterior pituitary gland, so the amount of LH decrease. When the amount of LH decrease, it cannot stimulate the Leydig cell to make more testosterone. So you see that the amount of testosterone decrease, and this is a system which we call it negative feedback. After the testes and all of this information about the testes, we have duct system. We have different ducts in the male reproductive system. I go back to this slide. I told you that this is a testis, and in each testicular lobule, we have 1 to 4 seminiferous tubules. As you see in this picture, the seminiferous tubules become straight in the last part of this testicular lobule. Let me mark it for you. This part which is straight, it's not coiled. This part is called a straight tubule. So after seminiferous tubules, we have a straight tubule. The straight tubules from all of the lobules combine together and make a network here, which is a retestis. And from the retestis, we have some ducts which go out of the testis. These ducts are called efferent duct. These efferent ducts combine together and make epididymis. So, first we have seminiferous tubules. After that, we have a straight tubule. Then we have retestis. After retestis, we have efferent duct, and the efferent duct can make epididymis. These are the duct system which you have in male reproductive system. The duct of epididymis is a very long duct. When it's uncoiled, it's about 6 centimeter. Sorry, it's about 6 meter. And when it's coiled, it's about 4 centimeter. The sperm pass these long ducts in 14 days, and during these 14 days the sperm can gain the ability to swim, and it can gain the capacity which we call it acrosomal reaction, and after that it can do fertilization. The acrosomal reaction means that in the acrosome, which contain many digestive enzymes, we have some changes. Our digestive enzyme become active, and now it can do fertilization. The epithelium which we have for this duct is pseudostratified columnar epithelium. But this pseudostratified columnar epithelium contain a very specific specialization in the apical border. The apical border of the cells contain sterocelia. Up to now, you learned about the cilia, which can beat forward and backward and help movement of material on the apical border of the cell, like the cilia which you have in the respiratory system, and I will talk about it in female reproductive system too. You learned about the microvilli, which increase the surface area, and this sterocelia is the other specialization which we have on the apical border. They are immotile long microvilli, and I will show it to you in the next slides. The epididymis is a comma-shaped tube. As I told you, it contains head, body, and tail, and it can store sperm for 14 days, for many, different months, and if we have ejaculation routinely, this sperm can pass this duct in 14 days. If we don't have any ejaculation, then the sperms which are located in these ducts are reabsorbed. Here you can see the cross section of epididymis. As you see here, it's a very coiled tube. And in the next slide, you can see it in larger magnification. In larger magnification, each cross section contains pseudostratified epithelium. As you see here, we have columnar cells, and the level of the nuclei in these columnar cells are in different level. So we call the epithelium pseudostratified columnar. And on the apical border, you have these long extensions. These long extensions are sterocelia. And inside the lumen, these parts are the sperms, which we have in the epididymis. So this is the epididymis, and the epididymis continue at the tail with vas deferens or ductus deferens. You see that these ductus deferens come up past the urogenital diaphragm, come to the body, pass both sides of bladder. Pay attention, it doesn't pass it from the middle of the bladder. This is the cross section. Pass from both sides of the bladder, go to the back of bladder, and then the duct of epididymis and the duct of seminal vesicle merge together and make ejaculatory duct. This structure is called a spermatic cord, and I will show you the picture later. So this is the vas deferens. This is the bladder, and you see that this vas deferens can pass both sides of the bladder. The ductus deferens can store and transport sperm. The dilated terminal portion of ductus deferens is called ampulla. And for the histology of ductus deferens, we have epithelium, which is the pseudostratified columnar. We have a thick muscularis externa, which contains 3 layers, and the last part is called adventitia. Here you can see the cross section of ductus deferens. The ductus deferens is a star-shaped tube, as you see here. The epithelium is pseudostratified epithelium. After that, we have lamina propria, and after lamina propria, you can see thick muscularis externa. This is muscularis externa. In this muscularis externa, inner and outer layer contain longitudinal muscle, and the middle layer is circular layer, and the last layer is called tunica adventitia. In this picture, you can see the posterior view of male reproductive system. The urinary bladder. These are the 2 vas deferens which come to the back of the bladder, and in this picture I want to show you this dilated part. This dilated part which we have at the end of vas deferens is called ampulla of the vas deferens. It's the ampulla of the vas deferens, and as you see here, this ampulla and the duct of seminal vesicle combine together, and the result is having this short duct on both sides. These ducts are called ejaculatory duct, and ejaculatory duct from both sides combine the prostatic urethra. In this picture, you can see the prostatic urethra, which is located in the prostate gland. Then we have the membranous urethra in the urogenital diaphragm, and the last part is the penile or spongy urethra in the penis. The ductus deferens can convey sperm from epididymis toward urethra by peristaltic contraction of the muscle. I told you that we have 3 layers of muscle: outer and inner longitudinal and middle circular muscle. The contraction of this muscle can make peristaltic wave, and this peristaltic wave help the sperm and the fluid to pass. This ductus deferens can store sperm for several months, and if we don't have ejaculation, reabsorption of the sperm take place in ductus deferens, as we have this reabsorption in epididymis too. And the last part which I want to talk about it today is the spermatic cord. Look at this picture. In this picture, you can see the structure of a spermatic cord. In the spermatic cord, we have different structures. You can see one pink duct. This is vas deferens. You can see one red structure, which is testicular artery. You can see a network of blue. This is the veins, the network of veins which we have around the vas deferens. This network of vein has a specific name. This name is pampiniform plexus. So pampiniform plexus of testicular vein can turn around this vas deferens, and they can regulate the temperature here. And you have a very thin green structure, which is the testicular lymphatic vessel. And all of these structures are covered by cremaster muscle, which is the skeletal muscle, and after that we have the skin. So this is the structure of a spermatic cord, and we have all of these parts in the spermatic cord. And here you can see the ejaculatory duct. These two dilated parts are the ampulla of the vas deferens. Here you can see the seminal vesicle in the back of the bladder. Their ducts combine and make ejaculatory duct, which enter the prostatic urethra. These ejaculatory ducts are very, very short ducts. The urethra is the last part of duct system in male, and it's a shared structure between reproductive system and urinary system. Both semen and the urine can pass through the urethra. It passes through the prostate gland, which we call it prostatic urethra, the deep muscle of the perineum, which we call it intermediate or membranous part of urethra, and the last part is the penis, which we call it a spongy urethra or penile urethra. The urethra is the last part. And in the next video, I will talk about the accessory sex gland and the penis for you.